Hello, my name is Mihaela Hajduk and today is January 21st, 2020, and today I'm gonna do the lab for aseptic technique and culturing the microbes for Bio 225. I already put my wash my hands, put my gloves, my mask, my safety goggles and my apron, and I wiped the surface with 10% bleach, and I already labeled my broth with S. epidermidis, with E. coli, and S. cerevisiae with the permanent marker. I use half a teaspoon of yeast in a warm water, and also with my marker I label the alcohol for my pipette, and I make sure the pipette is clean and the alcohol goes in the pipette. And here is my lighter. Here's my candle, and I'm gonna light the candle to start my experiment. So I'm gonna grab the vial and I'm gonna sterilize. And also I'm gonna sterilize this one. And with the pipette, make sure the alcohol is okay. I'm gonna grab 0.25 broth, place it here, and shake it until it's getting dissolved. I think I'm gonna add a little bit more of broth. I'm shaking until it's dissolved. Just a little bit more. And place back in a tube. Nebra. Okay. I'm gonna sterilize the tube. And start again with the E. coli. I'm gonna sterilize the tube. And make sure the all alcohol is out from the pipette. And same, put 0.25. And shake it until it's dissolved. It's not dissolved yet. Okay. Place it in the tube. Okay. Now sterilize again the tube. And put the cap on. Now I already made the yeast with the warm water, and it's for the S. cerevisiae. I'm gonna sterilize the tube. Also add 0.25 ml. In a broth. And sterilize the tube. Okay, and I have the vials and everything. I have already my bucket here with the bleach. I will dispose everything over there. Waited for 10 minutes, and after that I'm gonna put in a trash. This is my my starting for my aseptic technique and culturing microbes. Mihaela Hajduk, aseptic technique and culturing microbes, results from 60 hours of incubation time. The results is because my liquid didn't settle properly, so I left another 24 hours. And I'm gonna start with E. coli. It is a clear liquid and a white substance at the bottom of the vial. Sorry, my camera. Okay, and the next one is the S. epidermidis. It is the liquid is cloudy and a very thin white substance is formed at the bottom of the vial. And S. cerevisiae, the liquid is partially cloudy and a thicker layer at the bottom of the base of the vial we can notice. Hello, my name is Mihaela Hajduk and today is January 24th, 2020. And today I'm going to do the aseptic technique and culturing microbes, transferring the microbes from the microtubes to the agar plates, and I'm going to transfer in each quadrant. I already have my inoculation loop in my alcohol. I have my candle to sterilize the vial. I'm gonna start with E. coli. I'm gonna sterilize the vial. And with my inoculation loop, I will safely go my base. And now I'm going to spread in the first quadrant. Before I close in the vial, I'm gonna sterilize the loop again, the vial again. And with my second loop, I'm gonna spread the microbes, what it is in the first quadrant, on the second quadrant. With my third loop, I'm gonna spread the second from the second quadrant to the third quadrant, the microbes. And for the fourth quadrant, I'm gonna spread the microbes from the third quadrant to the fourth quadrant. And I finish with E. coli. Now I'm gonna start with the same process with the S. cerevisiae. Go in. And spread in the first quadrant. Okay, I'm gonna sterilize the vial and close it. Second loop, I'm gonna spread the microbes in the second quadrant. Let's see here. Okay. And the last quadrant. Okay, and I'm done with the SRVCA. Now I'm starting with the epidermidis. Same process. Okay. Now I'm gonna go to second quadrant. Now we're gonna go to the third quadrant. And the fourth quadrant. Okay, and close. I'm gonna sterilize the vial and close it. I'm gonna let the agar plate to rest for 48 hours and we're gonna see the results. Mihaela Hajduk, January 26, 2020, Aseptic technique and culturing microbes. These are my developed colonies on my agar plates, and I have for incubation for 48 hours. I'm gonna start with S. epidermis. S. epidermidis, the shape appears to be rhizoid. The margins are entire. The elevation is slightly raised and the size is punctiform. Next is the S. cerevisiae. The shape is circular. The margins are curved and the elevation it is convex in the size and is small. For the E. coli, it's hard to visualize, but it's there. The shape is irregular. The margins are lobate, and the elevation is flat, and the size is the pinpoint.